Now we are live. We'll just wait for a few minutes so that some audience can join. We'll wait like for 2 minutes or something, then we can continue. Okay. Okay, we'll start then. Hi everyone, welcome all to the fifth chapter of Biobattalion webinar series. Today with us is a remarkable and distinguished scientist, Dr. Babak Momeni, Assistant Professor of Boston College. Before we start, let me take the liberty to speak a few words about you, sir. Dr. Babak is trained as an electrical engineer. He attended Sharif University of Technology, Iran, where he received his bachelor's in electrical engineering and a master's in microwave and optical communications. He then moved to Georgia Institute of Technology, Atlanta, where he received a MS in physics and a PhD in electrical engineering in 2007. His doctoral training at the Georgia Institute of Technology involved designing and implementing compact on-chip photonic demultiplexers. He continued his work at Georgia Tech as a postdoctoral fellow to build spectrometers for biosensing applications. Intrigued by biological systems, he joined Dr. Shaw's lab at Fred Hutchinson's Cancer Research Center, Seattle, Washington, in 2009, where he studied the stability and spatial organization of microbial communities in synthetic communities. He joined the Department of Biology at Boston College in 2015 as an assistant professor. Dr. Babak and his team combine theory and experiments to study microbial communities that affect our health and environment. His current lab projects include examining the stability of microbial communities, characterizing and controlling microbes in human nasal cavity, and repurposing bacteria and their enzymes for efficient removal of food contaminating toxins. Sir, it's a pleasure for us that you took the time to be here with us today. Also, for the audience, if you have any doubts or questions regarding the presentation, please post your questions in the comment box and I'll be moderating it at the end of the presentation. So I welcome you, Dr. Babak, to the fifth chapter of BioBattalion webinar series. Over to you, Dr. Babak.
Thank you very much for the introduction. It's my pleasure to talk about some of the research that's going on in our lab with you all. So today I'm going to do more of a survey of different types of projects that are happening in the lab, and all of them come within this umbrella of harnessing microbial potentials. Before starting, I would like to thank people in the lab who actually did most of the work. These are postdoctoral researchers and graduate students, as well as several undergraduate researchers who contributed to this work. When we are talking about harnessing microbial potentials, the idea is that there are these microbes around us that are very diverse with many different metabolic capabilities, and they are present in several different environments. And that makes it likely that if we are looking for solutions to some of the current challenges, we can potentially find the solutions already within the microbial world. In our lab, we are in particular focusing on two directions. One is microbiota-based therapy, and by that I mean the use of microbes to prevent or treat diseases. And the second direction is microbial bioremediation, which means the use of microbes to remove waste and toxins. And I should give you this disclaimer that there is a lot of breadth in this presentation, but not a whole lot of depth. So if anything is unclear, please feel free to ask questions, and I'm happy to clarify things. So we'll start with microbiota-based therapy. And the idea comes from this observation that microbes that are associated with us offer several benefits, and that includes modulation of our immune system so that it can does its job properly, as well as protecting us from pathogens. There are additional benefits. For example, they help us in digestion or provide some of the vitamins as well. And because of that, disruption of these microbiota can lead to diseases. And examples of that are things that happen with eczema or colitis. But if we accept that as a fact, then we can also potentially intervene and manipulate the microbial community to prevent or treat some of these diseases. So that's the advantage of manipulating microbiota. But there is an additional advantage that it reduces our reliance on antibiotics, because if we are using other microbes to treat infections, we don't need to use antibiotics. And that's helpful because currently we are facing the rise of antibiotic resistance, which can be a major challenge in the coming years. But if we want to do microbiota-based therapy, what are the challenges? It sounds like an easy task. The main challenge is that microbiota are complex ecosystems. There are a multitude of species present. There are several unknown interactions, either among microbes or between microbes and their hosts, that are not characterized and not well understood. And the environment is complex. It's temporally and spatially variant and heterogeneous. So it's really hard to know what is actually happening in that environment if we want to control it. Another challenge is that the rules for intervention are not completely understood. So there are some intervention strategies that are currently being used. For example, probiotics are being used as a way of bringing back the microbial community to health. Or there is fecal matter transplant that in some cases of infection seems to be very beneficial. But in all these cases, we don't fully understand what is happening. And because of that, there are many cases that probiotics don't work, or several cases that fecal matter transplant has unintended consequences that we didn't plan and could potentially be really harmful. So there are even cases of death after fecal matter transplant. And we think the reason for that is that these are only partially successful because we don't fully recognize and appreciate the complexity of the situation. Then, faced with those challenges, what are the things that we can do? So the approach that we are taking starts with formulating predictive models to capture the properties of interest within microbiota. And the idea is that if we have a predictive model, then we can start from there and navigate the system towards the state that we are more interested in. The second step that we have taken is to find a system that's not too complex and perhaps not too simple as a way of allowing us to do mechanistic studies in microbiota. And the goal of this step would be to ensure that the models that we are using are realistic, and also to test and validate the hypotheses that we come up with. And then the third step is to search for general principles that can guide intervention strategies. So rather than focusing on specific cases and specific situations, we are looking for principles that could inform and inspire how we should intervene and modify microbial communities. So let's get started with the modeling part. For modeling communities, the traditional approach has been to use Lotka-Volterra type models. And these are models that have a long history of being used in the context of ecological communities. They are relatively simple in the sense that regardless of the mechanism, they abstract all the interactions into a fitness effect between different species. Let me explain what I mean by that. So let's assume that you have two species. These types of models just tell you that depending on how many of species one you have present, how much species two benefits from them, or depending on how much of the species two are present, how much species one, for example, gets inhibited. And the history comes from the interaction between lynx and hare and the prey-predator interaction that was there. So these models have a long history, and they have been successful in capturing some of the dynamics that happen ecologically. So in this particular case, there was data from 1840s to around 1930s that showed oscillations between populations of hares and pigs. And this simple Lotka-Volterra type model was capable of capturing these oscillations. So people got really excited. It was a really simple model, but it was capable of giving realistic predictions about how the populations would fluctuate in nature. So there are several advantages. One is that there is no need to know interaction mechanisms when you're forming Lotka-Volterra type models. They are easy to estimate parameters. All you need to do is to measure population densities, which is a fairly easy thing to do, especially these days using sequencing. You can easily measure the population composition. There is some empirical support for it, so there are cases that they have worked well, and they are easy to extend to multi-species communities. So all you need to do is to take pairs of species, characterize their interactions, and do that for all different pairs within the community, and then put all of those models together and form a network of interactions that describes the community as a whole. So this all sounds good, but does this type of modeling apply to microbial communities? So the tradition is in macroecology among larger species. So to answer that question, we started from a simple thought experiment. We said that if we have a ground truth and we know exactly what happens within the system, and if we formulate a pairwise model of Lotka-Volterra type as an approximation for that model, and we simulate the dynamics using both models and compare predictions, do they agree with each other or not? And what are the conditions that they agree and what are the conditions that they don't? So this line of questioning tells us whether this type of model could in general work or not. And instead of using a realistic biological system, we are using a mechanistic model as our reference, because in this case, we know exactly what is happening within the community, and we are not bound by experimental errors or measurement restrictions. So we did that, and for our mechanistic model, we formulated a mediator explicit model. And by that, what I mean is that instead of only looking at interactions between species, we also included chemicals that mediate those interactions. So these chemicals could be metabolites or toxins or antibiotics. And in our model, we assume four simple types of interactions. The type that we show as an arrow up here tells us that the species S is removing chemical C from the environment. So this could be, for example, consumption of a metabolite. This type of arrow shows that species S is producing a chemical in the environment. So this could be something that gets secreted, or even potentially something in the environment that gets converted into C. And then in return, chemical C can either stimulate the growth of S, or it can inhibit the growth of S. So these are fairly simple types of links, but they capture the majority of cases that we can think of that species are interacting through chemicals or metabolites in their environment. And then we take these links, put them all into a single model with a set of species and set of chemicals, and each of these species produce or consume a subset of chemicals, and each of the chemicals either positively or negatively affect a subset of species. And that's our ground truth as our reference model that could represent a realistic microbial community. And then we asked a few simple questions. First question was, can the Lotka-Volterra type model represent different types of microbial interactions? And for that, we looked at two simple types of interactions. One that we call reusable, in the sense that the chemical is reusable in this interaction. Species 1 is producing a chemical, species 2 is getting affected by it, but the species 2, which is the recipient, does not change the concentration of chemical 1. So it's not consuming it or removing it from the environment. In contrast, you can have consumable interaction, that one species is producing a chemical and the other species is consuming it and at the same time getting affected by it. So an example of reusable interaction could be maybe when species 1 is changing the pH in the environment, and that change in pH can affect the species 2. And in the second case, we can have a species 1 producing a metabolite, for example, an organic acid, and a species 2 is consuming that organic acid and maybe benefiting from it. And what we saw was that if you formulate the model, a pairwise model for these cases, you end up with two different types of equations, and these two types are not interchangeable, in the sense that if you are simulating a reusable type of interaction, you can't use equations for the consumable type of interactions, and vice versa. And what that tells us is that the simple local type model, which is shown here, cannot capture different types of microbial interactions. Even something as simple as a metabolite in the environment that gets consumed is not properly approximated in this model. And if we look back, this is almost trivial. In this case, if species 1 has produced a chemical, it gets distributed among recipients. Whereas in the other case, it affects all the recipients without getting distributed among them. So you would expect that the number of recipients does not affect the interaction in this case, and it affects the interaction in this case. So we naturally would expect that these two would not be consistent, and that the local type model is not adequate for capturing other types of interactions. The other question we asked was what happens if we go to more than 2 species? And we put together simple communities to see the actual situation. In this case, we have 3 species that are interacting through a single mediator. Species 1 is producing it and getting inhibited by it. For example, if it's acetic acid, we can have this type of interaction. The species 2 consumes C1 and benefits from it, and the species 3 is just being inhibited by it. So if we do pairwise interactions, we can come up with this type of network connectivity. For example, in the absence of species 1, there is no chemical 1, species 2 and species 3 are not interacting with each other. But when we simulate the dynamics using the original model shown with solid lines here versus the local type approximate, which is shown by dotted lines here, we see that the dynamics are very different. And I would like to emphasize that in this case, each pairs of interactions work pretty fine. So if we measure the deviation between the local type model and the mechanistic model, all the pairs S1, S2, S3, S1, and S2, S3 are offering good approximations. But when we put the 3 of them together, they are no longer a good approximation. And the reason for that is the presence of S2, for example, is affecting the interaction between S1 and S3, because it's removing the mediator from the environment and weakens the interaction between S1 and S3. And that's an effect that's not included in the local type model. In the local type model, we are assuming that all interactions are pairwise and independent of each other. So another failure of the local type model. So what should we do? We know that many interactions among microbes are happening through the chemical environment. So should we just abandon local type model and move on with our lives? Or maybe there is more to it. So when we look at the literature, there are several examples that show that the local type models appear to work fine. There are cases with community assembly or some synthetic communities of gut microbiota that show that local type models work just fine. But there are other examples that show that they are not enough. There are examples with, again, analysis of gut microbiota data that shows local type model is not good enough. And people have also done some controlled experiments, and they see that local type model doesn't always work. So we changed our approach and asked this question: that what are the conditions under which these simple local type models work? So answering this question would be very, very beneficial, because it tells us where we can use the simple model and where we cannot use it, and we should look for maybe more complex models that capture the actual dynamics. And to answer this question, our solution is to test our ideas in a controlled biological relevance setting and see in a situation that we can control the microbial community and grow them in a lab environment, and we can reliably measure them, when and why local cultural type model would or would not work in that situation. So the system that we picked for this study is the nasal microbiota. And our main motivation for it is that nasal microbiota has an important impact on our respiratory health. It's been observed that nasal microbiota can prevent colonization of opportunistic pathogens. And an example of that is Staphylococcus aureus, which is one of the members of the community. In general, around 30% of the population have Staphylococcus aureus in their nose, and even around 2% carry the antibiotic-resistant version of it in their nose. And for most people, this is harmless. They are present in that environment, but they don't cause any infections. But if the whole host immunity gets weakened, or the patient is dealing with other illnesses, then Staphylococcus aureus can become pathogenic. It can cause bacteremia or blood infection and infection in several other organs. And that's an important infection type because it has been shown that it caused around 120,000 invasive infections, most of them in the hospital setting, and more than 20,000 deaths in the US. It's a global issue, but unfortunately, there are not reliable numbers throughout the globe. Interesting for us, it's been observed that other microbes that are present in the nasal cavity environment can control the colonization of staph. So the 30% that have the staph persistently in their nose versus 70% that they don't have it, the thought is that in those 70%, the presence of other microbes has prevented the colonization of Staph aureus. And that brings this idea that can we restructure the community, maybe even using the members that are already present in that environment to reduce the load of Staph aureus and reduce the risk of people getting infected with their own microbes. As an additional bonus, nasal microbiota has a relatively low diversity, which makes it a good system to study in the lab. So if you sample the human population, around 20 species explain the majority of the reads, more than 90% of the reads that come from sequencing. And 19 out of those 20 are easily culturable in the lab, which means we can reliably capture most of the things that happen in the nasal microbiota. Even more, if you look at individual persons, you see that in each community, somewhere between 3 to 8 species are dominant. So each of these microbial communities are not really that complex. They have only a handful of species present, which makes it a simple model to work with in the lab. So we have taken isolates from the nasal microbiota, and the isolates that we took was based on the data that's available in terms of what species are dominant in that environment. So these graphs show the genera that explain the majority of abundance in the sequencing data. So you see Corynebacterium, Cutibacterium, and Staphylococcus are the major genera in that environment. And if you look at the species or supraspecies level, you see some of these species that show up as very prevalent within the nasal microbiota. So because of that, we collaborated with the Lemon Lab, now at Baylor College of Medicine, and they were kind enough to give us some of these species that are clinical isolates, and they have come from healthy individuals, but they also represent some of these species that are highly represented within the nasal microbiota. So we took those species and we studied them in the lab. By studying them in the lab, I mean we grew them and looked at their growth properties, but we also looked at their interactions with each other. And to characterize these interactions, we used a simple type of experiment. We call it cell-free spent media, or CFSM. in which what you do is you grow individual species in monocultures, you centrifuge them to remove most of the cell, and then filter them out to have cell-free spent media of each of the species. And then you expose other species to that cell-free spent media and ask how well do they grow in this new environment. And that will tell us something about the metabolite-mediated interaction between different species. If a species one has maybe removed one type of sugar from the environment, then you see how species two responds to the depletion of that sugar. Or if species one has produced something, some byproduct of metabolism as part of their regular activities, for example, some organic acid, then you can see how species two responds to the presence of that organic acid in the environment. So what we do is that we systematically do this for all pairs of a species and measure growth rate and carrying capacity of each of these species in the cell-free spent media of other species. An important thing that we observe just based on the equations is that if you do this, the ratio of growth rate to carrying capacity in spent media turns out to be equal to the growth rate, the ratio of growth rate to carrying capacity in fresh media if Lotka-Volterra type model is true. And that gives us a simple test to ask whether Lotka-Volterra type model is a good approximation or not, because we know that if it is true, when we do these experiments and calculate these ratios, they should stay the same. So just to reinstate it, if Lotka-Volterra type model holds, the ratio of growth rate to carrying capacity in cell-free spent media or supernatant assays is independent of the species that are used for this for getting this CFSM. And Sandra, a graduate student in the lab, did all these experiments of looking at pairs of species, and she saw that in many cases we observed this trend to be true. So if you look at R-squared values, they are pretty high. There is one exception because there is an outlier in this case. But overall, it seems that in many of these situations, when we look at interactions between nasal bacteria, the ratio of growth rate to carrying capacity seems to be a constant regardless of what supernatant we are using. So we started from this point and asked first, maybe does the resource availability explain this trend? And we did the experiments in low nutrient environments first. So we diluted a typical growth media for these species to quite a bit. So starting from 100%, we went to 5% all the way down to 0.32%. And at these low concentrations of the media, we saw that the trends still would hold. So what this means is that if you take the media from one species and you just dilute it, both the growth rate and carrying capacity will scale the same way. So this suggests that maybe resource availability could be a good explanation for why we see the same trend when we are looking at interactions. It's simply one species removing some of the resources from the environment, and the other species has to grow in the lower nutrient environment. So it just follows this trend. Importantly, when we did the same experiments at high nutrient concentration, so this is going from 10% THY all the way to 100% THY, the linear trend did not hold anymore. So whatever trend we are seeing applies to low nutrient environment, but not to high nutrient environment. We took one more step and asked if we change individual metabolites or mediators in the environment, how does that affect the growth of different species? So here I'm showing you a subset of data with some of the compounds that we tested and a few of the species. And as you can see, the response of individual species to the mediators is not necessarily the same between different species. That maybe if you try acetic acid, Staph aureus gets affected by, and it doesn't grow as well, but Corynebacterium tuberculostericum does not get affected by pH much, or by acetic acid much, so it stays there. In contrast, it gets affected by pH, but maybe Staph aureus doesn't get affected by pH. And overall, we saw that the linear trend did not necessarily hold for individual compounds. So if you modulate one of the compounds in the environment, that doesn't guarantee that the Lotkov type model would still hold. So we saw that the responses were compound specific. So it was different between, let's say, acetic acid and vancomycin, which is an antibiotic. And it was also species specific, that not all species responded the same way to different compounds in the environment. So from these observations, our model is that in a low nutrient environment, a combinations of effects that could come from removal or addition of resources in the environment, or removal or addition of some of these additional metabolites in the environment. The effect of all of those combined on average creates a new habitat in which the other species can grow, and that effect falls on the linear trend. So from this observation, we went back to the literature and asked whether this observation has support in what people have already explained. And we found this 2015 paper from Lipson. And what he was doing was that he was trying to explain inconsistent relationships that have been observed in terms of rate yield or rate carrying capacity in previous data. So people had done different experiments, and they had seen that in some cases they would see positive correlation between rate and carrying capacity, and in some cases they would see negative correlation. So what he did was that he tried to capture different situations and came up with this theoretical model that has two different zones. So in low nutrient environments, he called it maintenance energy zone, that he thinks that in this case there is a positive correlation, but if you go to high nutrient environment, there is a negative correlation. And I'm oversimplifying things, but in effect what he's saying is that in low nutrient environments, because the nutrients are so low, any nutrient will be used for producing new biomass, and that contributes equally to growth rate and carrying capacity. And because of that, you see a positive correlation between growth rate and carrying capacity or yield in low nutrient environment. In contrast, in nutrient-rich environments, the cell can pick different strategies. So the cell can grow really fast, but be a little bit more wasteful, or it can grow more slowly, but take advantage of all the resources in the environment more effectively. And because of that, there is a trade-off between how fast cells grow and how much cells they produce, or because of that, you see this negative correlation between growth rate and carrying capacity. And what we have seen in our experiments is consistent with this view, and importantly, in the range that we are interested in, based on the amount of nutrients that are measured in the nasal microbiota environment, we know that we are in the low nutrient environment. And because of that, we think that Lotkov type model could work fine for these types of communities. Moving on from that point, we searched for general principles, and one of the principles we were interested in is this idea of coexistence. So when we're talking about some of the diseases, it's come from dysbiosis, that some of the members that are usually present in our microbiota, if they go away, then we will face some of the consequences. So we asked ourselves, what keeps them together to begin with? What allows them to coexist with each other? And that's the main question that we were trying to answer. What allows interacting microbes to coexist, even though on their own they have very different growth properties and needs? And the approach that we took was that we used the model of interactions through metabolite mediators. This is the same mechanistic model that I introduced earlier. And we start from an initial pool of microbes that are randomly interacting with each other, and we simulate those communities over rounds of growth and dilution until we end up with an enriched community that has maybe fewer species, but those species are stably together. And we repeat this process thousands and thousands of times. And the idea is that if we look at the properties of initial pools of microbes and properties of the final communities that get enriched, then we can get some ideas about what influences coexistence. So the main trends that we observe come from this capability to do many different simulations of the order of tens of thousands of simulations and look into this large data set to come up with trends. This is something that you can't easily do experimentally just because of the amount of effort that it takes. And from these experiments, the things that we've learned, I'm showing one of the examples of the graphs, but there are a few things that I want to emphasize. One observation was that so here we are simulating an initial pool of 30 species and 10 mediators, and simulating how the richness, what's the frequency of observing communities of different richness in the end. And first, let's compare the graphs on the left with the graphs on the right. So these are the same colors represent the exactly same community with one difference. In this case, we have the majority of interactions to be facilitation or positive influence of species on each other. In this case, we assume that most of the interactions are negative. And you can easily see the difference. So in this case, it's very unlikely to come up with a community that has high richness, whereas in this case, you can find communities with high richness relatively easily. So with relatively high frequency, let's say 10% or so, you can find a community of richness 4, which means there are 4 different species coexisting with each other. Another aspect that I wanted to emphasize is that within these examples, we are also changing the ratio of average consumption to average production of different metabolites. And you can see that as we go to higher and higher ratios, the richness in the communities increases. So for example, in this case, as we are increasing, so as there is more consumption and less production, we end up with higher coexistence. And our interpretation is that first, interspecies facilitation allows improved coexistence. And we see that clearly by contrasting the left-hand side with the right-hand side. And we also see that consumption of beneficial metabolites regulates the balance of a species, because one species, if it's benefiting from something and it's consuming it, it's also removing that benefit. And that acts as a balancing force to keep a species together. The other question that we tried to answer was the idea of colonization resistance. And that means the ability of a species, microbiota, to prevent the colonization of invaders or pathogens. And this is very important for our health. We use probiotics or microbiota transplant, for example, fecal matter transplant, in this context, that there is resistance from the microbiota to accept new species. And we, in some of these cases, we want to introduce a new species, or in other cases, we want to prevent the introduction of new species, for example, a pathogen. And we ask how interactions among species contribute to the success or failure of invasion. And to answer that question, we came up with this conceptual assay to categorize different types of outcomes that we can get. So we are starting from a resident community at the beginning. We are introducing an invader into that community, and we are asking whether the invader is maintained in that environment or it drops out, and whether the resident community remains intact or it loses some of its members. And based on these 2 axes, we define the outcome. So you can have augmentation, for example, if the new invader becomes part of the community and everything else stays there. Or you can, for example, have resistance if invader goes away and the initial community maintains its diversity. You can also have displacement, which one or more of the initial species get replaced by the invader, or we can have disruption, that the invader knocks out some of the resident community, but it's not capable of staying in that environment either. So what we did was that we simulated again many different examples and quantified the fraction of these outcomes. How often, for example, we see resistance versus disruption versus augmentation versus displacement. So I'm showing the frequency of these outcomes over some parameter of interest that we studied. In this case, we are looking at the propagule size, which is the number of invader that we have introduced into the community. And importantly, what we see is that for a wide range of propagule sizes, there is no real change in the outcomes. And that's something that we have to consider when we are considering intervention strategies. For example, if you're introducing a probiotic, you want to uh, have more augmentation, but just adding more of it up to really large numbers, like 20% or so, which is huge considering how much microbes you can practically introduce. So up to that point, there is no real benefit from adding more and more of the probiotic. And only at really high concentrations you see an effect, but even in that case it's not contributing to augmentation. Instead, it's displacing or disrupting the community and losing resistance rather than introducing the new member. As another example, we again simulated many cases but removed different types of interactions, and what we observed that was that the mutual beneficial interaction between the invader and the resident community is the main factor that determines the outcome of augmentation. So if you want augmentation, instead of trying to add more and more numbers, you have to focus on interactions and create a situation that both the probiotic that you're trying to introduce and the resident microbe benefit from that interaction. So to summarize everything so far, in this context of microbiota-based therapy, we have used in vitro nasal communities along with corresponding mathematical models as a tractable system to formulate and test hypotheses. And hopefully that would help us design future intervention strategies, because these are systems that we can easily control. We found that simple low-level type models might be adequate, but only if the environment is nutrient poor, so it's relatively low in nutrients, and it's complex, that there are multiple interaction types that are happening in that environment. And we also searched for general principles, and we came up with some ideas that could help us decide what to do to change coexistence or colonization resistance. So now I switch gears and talk about the other project in the lab that I wanted to share with you. And this project is about bioremediation. And the context is that we want to remove some of these contaminations that affect the food that we consume. The types of contamination that we focus on are mycotoxins. So these are fungal secondary metabolites that contaminate a lot of different resources and are really harmful for consumption. They are fairly widespread. They contaminate around 25% of world crops, and they cause a lot of damage to the body, from inducing new mutations to cancer to tissue damage, for example, liver damage, to immune depression. And it costs somewhere around $3 billion to farmers in the US to get rid of these compounds, or sometimes because they have to throw out the stock that gets contaminated. I want to emphasize that this is not just a conceptual concern. There are strict regulations by FDA that controls the quality of food, and once in a while there are alerts that some stock is not safe for consumption and it has to be removed from circulation. Here in the US, it mostly affects farmers and food producers, because if their stock is contaminated, they can't sell it anymore. They can collect it back from the market and destroy it. In low-income countries, these regulations are not enforced as much, and that means some of these toxins go through the food chain and people consume them. And as a result, there is around 4 times higher rate of liver cancer in developing countries. And liver cancer itself is the second leading cause of death. So it costs communities even more because of the health consequences. And even in Kenya, not too long ago, there were cases that the amount of contamination was so high that it even led to direct death because of consumption of contaminated corn. There are approaches to remove these contaminations from the environment. There are physical and chemical approaches, but they are relatively expensive, they are not reliable, and they might also affect food quality. So food producers are not keen on using these approaches. There has been suggested that biological detoxification could be a promising alternative. So use of biological organisms or their enzymes to remove these toxins from our environment or food. And the main challenge is that the things that we have found so far in nature are not good enough for the applications. So they are not fast enough in removing the toxin, or they might have side effects, or they are not safe to introduce into the food. And we think that the true potentials of microbes is not still known because we haven't really sampled enough. And because of that, so this is the area that we thought we could potentially contribute. The main idea is that to take something that the nature is already offering us and build upon it to improve the detoxification performance. So take a simple example of detoxification by an extracellular enzyme. So there is an enzyme in some host organism, it's potentially regulated, it produces the enzyme, that enzyme potentially gets secreted, and that could also be regulated. And then outside the cell, it takes the toxin and makes it less toxic. So if you think about this overall picture of how detoxification is taking place, you can think about how to improve it as well. So one approach is to just find better host organisms, things that have better enzymes or produce them more or secrete them more. And we can do that by screening for species and mutants to find better detoxifiers. Another approach is to engineer better detoxifiers. So you could take a host organism that already has some of these capabilities and genetically engineer them, for example, overproduce the enzyme or overexpress the secretion machinery to make the overall process more effective. But this requires us to know what exactly is happening within the cell. And the third approach is to take the enzymes, so rather than focusing on the host itself, take the enzyme and modify it to improve its performance against the target mycotoxin. So we have done a little bit of work in each of these directions. For finding better detoxifiers, a graduate student in the lab, Natalie, has looked at screened several strains from our environment and asked how good they were for detoxifying aflatoxin, which is one of the major categories of mycotoxins. And interestingly, she found that she was able to find many different isolates that showed detoxification. And she did this on glucose and starch for isolation, and also tested their performance on glucose and starch as the main carbon source in the environment. And the major thing she found was that strains that she isolated on glucose and then exposed to starch when she was characterizing their performance showed the best performance. And we think the reason for that is isolation on glucose is less restrictive, so it allows us to get more of the species in our environment in our screen. But when we are testing them on a starch, the presence of a starch in the environment triggers these cells to produce more of the enzymes for complex carbons, and some of those enzymes degrade the mycotoxin of interest. And then another important aspect is that based on our observations, the detoxification of aflatoxin is not a rare event. We can easily find many species that show really high performance for detoxifying aflatoxin using a simple screen. And that's something that wasn't appreciated before in the literature, that they had found examples of different species, but the prevalence of aflatoxin detoxification wasn't known and appreciated. We are also using droplet-based screening as an alternative to find good detoxifiers. So the idea is that instead of putting individual cells in large-scale wells, we can put cells inside droplets and watch how they perform. And in our case, because the toxin is fluorescent, we have an easy assay to watch the fluorescence and decide whether the cell is doing a good job or not. And you can see that these droplets that have cells in them also have a drop in fluorescence, which means they are effective in removing the toxin from the environment. But these types of screens allow us to go at a much higher throughput We can potentially even screen thousands of droplets per second, and this allows us to um, screen several many mutants and find out what types of genes might be involved in the process. The main limitation is that we need fluorescence for our assay, but for for several aflatoxins, that's not a restriction really. We also looked at the enzymatic capability of microbes. So we asked, can we figure out what the mechanisms are? And in this assay, we started from cell-free filtrates of Rhodococcus species, which were known to have aflatoxin detoxification capability. And we, unlike our expectation, we saw that instead of the enzymes completely depleting the aflatoxin, they slowed down and eventually stopped. And we used a simple model of assuming that there is decay in the enzyme itself, and that simple model shown by dotted lines here seems to capture the actual dynamics pretty well. So we think what's happening in this environment is that instead of the enzyme constantly removing the toxin, it loses its activity over time. So we compared different species of the Rhodococcus, our Peridinivorans and our Erythropolis, and we saw that there was a difference in their enzymatic activity at the beginning, and also a difference in their in the lifetime of their activity. And you might expect it or not, but there's also seems to be a trade-off that the one that has lower activity has higher lifetime. So we did this for a whole bunch of cases for both glucose and starch environment and for Predinivorans and Erythropolis species. And what we saw was this consistent trend between them, that there seems to be an intrinsic trade-off between them, that the decay rate of the enzyme is proportional to the initial concentration of the enzyme in the environment, or initial activity of the enzyme in the environment. And this trend doesn't seem to be strain dependent or condition dependent. All of them sort of more or less fall on the same line. And that tells us that there is, if we include this restriction in our model, we see that there is a limit on how much of the toxin these species can effectively remove from the environment. And the toxin concentration cannot be too high, because even if we use too much of the enzyme, the life of the enzyme drops, so it ends up not doing as good of a time. And there's ongoing work that we're still trying to figure out why this trade-off exists and what is the cellular machinery that affects it. We also looked at the enzyme itself. So we collaborated with a lab in Europe that they do larger scale molecular modeling using quantum mechanics. So we simulated one of the examples of enzymes, lacase from Dermatis versicolor, which has shown to have the capability to degrade aflatoxin. But when we tested this in the lab, we saw that two different versions of aflatoxin, aflatoxin B1 versus aflatoxin G2, showed very different performances. And this is something that existing models can't explain why this difference exists. So we got a little bit deeper into the quantum mechanical modeling and tried to figure out what might be the reason for that. And what we learned from this process was that first, the detoxification itself takes place by lactone ring opening, which is this ring over here, following an oxidation event. So lacase, what it's doing is that it's oxidizing the molecule, and that makes it unstable and ends up breaking down the molecule and making it not toxic. The other thing we learned was that the strength of this oxidative capability is not the rate limiting factor. So it's not like a lacase that's better at oxidizing the substrate will do a better job. Instead, it seems that the mismatch between the aflatoxin and the enzymatic pocket is the rate limiting factor. And we also observed that there is a difference between AFB1 and AFG2. AFB1 only essentially loses electron from one end of the molecule, whereas AFG2, there are several places on the molecule that can lose an electron. And we think that this gives this AFG2 more flexibility so that even if it doesn't completely fit within the enzymatic pocket, it might still lose the electron and get oxidized. And then we also observed that there might be some non-specific binding of AFP1 to lacase away from the active site that might be responsible for the difference that we observed. But this is still an ongoing work and we are trying to figure things out. So to summarize this last part, there are existing mycotoxin detoxifiers that show some potential, but they are typically not enough for the current needs in the food and feed industry. And there are opportunities to improve them. We can screen for better species or screen for mutants and find the genetic roots of detoxification. We can potentially engineer the strains to be better at detoxification, or we can take their enzymes and think about what changes in those enzymes could make them better at detoxifying the aflatoxin. And that's the conclusions. I'll leave it here for you to read, and I'm happy to answer any questions that you might have. Thanks.
Great. It was indeed an interesting and wonderful presentation, Dr. Babak.
Thank you.
I'm sure that all the audience watching this would be benefited from your talk. So we have got few questions here. First question is from Harshak Rajachandran. How about studying the interactions of a multi-species interactions using the Lotka-Volterra model?
Right. That's a really good question. It's an ongoing research in the lab that we are trying to see how far we can expand it. So far, it seems in low nutrient environments, things still work pretty well, but it's something that we have to confirm. And we are doing those experiments using nasal bacteria.
Next question is from Vaishya K. Vishwanathan. How effective is fecal transplant therapies under these theories?
So the issue of fecal transplant is a little bit of a black box. So usually the screening that goes into it is only to screen whether there are pathogens present or not. So if pathogens are present, they won't use that. But other than that, there is not a whole lot of information about what's actually in that environment. Some thoughts are that metabolites in that environment are making an impact. Some thoughts are that there are some of the species that make an impact, and probably the reality is that it's a combination of both. But we don't know about that system to apply any of the theories yet. So there is a gap between what we know and what we can design. And right now, the things that we know about fecal matter transplant is so little that we can't effectively design anything.
Next, we have a question from Vaishak. Out of curiosity, how was your switch from engineering to biology?
This is,
A
I guess, one of
follow-up
those
question is there. What was your motivation and what were the challenges?
things. So I guess to completely be honest, initially when I made that transition, the thought was that I need to learn more about biology. So I went to a lab to do that, and then I was thinking maybe naively that I will learn something about biology and I go back to technology, which was my comfort zone. But when I started working on these synthetic systems and simple microbiota, it was really interesting, and I sort of got hooked in there, and then never really left that field. The main challenges is sort of not having the background that standard biology students have had during their normal training. So it's a little bit challenging to go into a new field and try to find your footing in that new field, and that's an ongoing challenge for me. There are still things that I wish I knew earlier that I don't, but it's part of sort of learning experience, and I've been enjoying that process.
Next question is again from Vaishak. Will multi-omics data help in modeling complex microbial interactions?
Well, the short answer is yes, definitely. The I think genomics has already taught us a lot about what's happening in these communities. Now the transition is to look at metabolomics and proteomics, which are a lot more complex, but also a lot more informative. So if we can crack, for example, how metabolites in our microbiota are affecting different species and different environments, then that could potentially open up new intervention strategies and new treatments as well.
Any more questions, audience? Next, we have a question. Since a huge percentage of microbiome resides in gut, how far have we come in using microbiome-based therapies in treating intestinal disorders or autoimmune disorders like Crohn's?
Um, so there have been steps, important steps taken, but it's not nearly um, enough to propose it as a treatment, especially for things related to autoimmune diseases. There are issues that remain unsolved. There are cases like C. difficile infection that have a pretty good success rate, but presumably that's because the normal gut microbiota gets completely switch during that process. During the infection, it completely switches out. And the thought is that once it's completely out of ordinary, then it might be easier to introduce new species into that environment. And that might be some of the reasons that FMT works fine for many of the C. difficile patients. But if the community is maybe a little bit disrupted, but not completely destroyed or out of ordinary, then it's harder to manipulate it as well.
Any more questions? Okay, I think that's it. We don't have any more questions. So it was an indeed interesting and wonderful presentation, sir. Uh, Thank you once again, Dr. Babak, for such an informative presentation. And Dr. Babak, after the live streaming, we'll meet back in the broadcasting studio. So I kindly request you to stay after the live streaming. Also, thank you, audience, for joining us today. Also, for the audience who didn't subscribe to our YouTube channel, please subscribe. To know about the upcoming events, please go to our website link given in the description box and register so that you will be notified. See you all soon in the next talk. And so we are going to end the live stream now.
Thank you very much.
Bye, everyone. Um,